Built from fat, updated architecture first outline. Introduction: the great biological misunderstanding. Modern nutrition did not lose its way through a lack of data. It lost its structure. Once the body was reduced to an energy processing system, food was stripped of its physical meaning and recast as interchangeable fuel. Fat, carbohydrate, and protein were assigned values based on combustion rather than function, and from that point forward, the material differences between them were treated as secondary. The body became an accounting problem. Inputs were measured, outputs were estimated, and internal machinery was assumed to behave predictably as long as the numbers balanced. That assumption removed the one variable that actually determines behavior, what the body is built from. The human organism is not defined by how much energy it processes. It is defined by how it is constructed. Every cell maintains a boundary that separates internal chemistry from external environment while allowing controlled exchange. That boundary is not theoretical. It is a lipid structure with measurable physical properties. Its composition determines how easily ions pass, how tightly gradients are held, how receptors behave, and how signals move across space and time. When the composition of that boundary changes, those properties change with it. Electrical stability shifts. Signal timing drifts. Transport becomes either restricted or excessive. None of this is captured by calorie models because energy does not define structure. Structure determines how energy is used, where it is directed, and whether it produces stable output or systemic noise. When lipids were reclassified as fuel, their role as structural material was ignored. Dietary fat became something to burn or store, not something to build with. This created a mismatch between input and requirement. The body continued to rely on lipids to construct membranes, organize signaling domains, and stabilize electrical systems, but the incoming material became increasingly inconsistent in quality and composition. Over time, this forces continuous remodeling under suboptimal conditions. Membranes lose coherence. Receptors fail to maintain consistent orientation. Signaling pathways activate without clean termination. The system compensates, but compensation has limits, and once those limits are reached, instability begins to spread across systems that appear unrelated but share the same underlying structure. Inflammation, in this context, is not an isolated response. It is the consequence of structural disturbance within membranes that alters how signals are generated and resolved. Hormonal instability follows the same pattern. Receptors embedded in unstable lipid environments do not respond predictably, even when hormone levels are unchanged. Metabolic dysfunction emerges when mitochondrial membranes lose the ability to maintain tight gradients, forcing energy production to become inefficient and inconsistent. These outcomes are often treated as separate conditions, each with its own explanation, but they are better understood as different expressions of the same architectural problem. Energy-based thinking cannot resolve this because it operates at the wrong level of the system. A calorie can describe potential input, but it does not determine whether a membrane will hold its structure under stress, whether a receptor will cluster correctly, or whether a signal will propagate with precision. It cannot explain why two individuals consuming the same amount of energy can produce entirely different physiological outcomes. Those differences arise from how the system is built, not how much fuel it receives. Reframing the body as a lipid-structured system changes how inputs are evaluated. Dietary fat is no longer a passive energy source. It is a material that integrates into membranes over time, altering their physical properties and, by extension, the behavior of every system that depends on them. Some lipids reinforce stability, resist oxidative damage, and support consistent signaling. Others introduce fragility, increase susceptibility to breakdown, and amplify noise within the system. These effects are not immediate in the way energy is. They accumulate through repeated incorporation and remodeling, gradually shifting the operating characteristics of the organism. This shift in perspective removes the need to treat symptoms as isolated failures. When structure is corrected, function follows without direct intervention at every downstream point. Membranes regain consistency. Signaling becomes coherent. Energy production stabilizes because the conditions required for it are restored. The body does not need to be forced into balance. It returns to balance when the materials used to build it support stable operation. The central error, then, is not simply that fat was misunderstood. It is that the body was mischaracterized as an energy system rather than a structural system. Once that error is corrected, the rest of the model begins to align. Food is no longer evaluated by how much energy it provides, but by what it becomes. Physiology is no longer explained through isolated pathways, but through the behavior of interconnected structures. The outcomes that once appeared complex begin to resolve into predictable patterns because they are governed by physical constraints rather than abstract models. The collapse of structure into calories. The reduction of food to calories replaced physical reality with abstraction. Once energy became the dominant lens, the material differences between fats, carbohydrates, and proteins were treated as secondary details rather than primary determinants of biological behavior. Combustion values became the organizing principle. For calories per gram for carbohydrate, 9 for fat, and the system was considered explained. This framing removed the question of what these substances become once they enter the body and replaced it with how much energy they could theoretically release. The body was modeled as a furnace instead of a constructed system, and in that transition, the role of structure disappeared from the equation. This shift did not occur because it accurately described physiology, but because it simplified measurement. Energy can be quantified cleanly. Structure cannot. Membrane composition, receptor organization, lipid domain behavior, and electrical stability do not reduce to a single number. They require a layered model that accounts for physical arrangement, material properties, and dynamic interaction over time. By abandoning that complexity, nutrition gains simplicity at the cost of accuracy. Foods that produce identical caloric values were treated as equivalent inputs, even though they deliver entirely different structural materials that integrate into the body in distinct ways. When structure is removed from a model, cause and effect become obscured. A system built from unstable lipid inputs will behave differently from one built from stable materials, even if total caloric intake is identical. Membranes assembled from highly unsaturated fatty acids exhibit increased susceptibility to oxidative damage, altered permeability, and inconsistent receptor behavior. These changes propagate outward. Ion gradients become less stable. Signal transduction becomes noisy. Mitochondrial efficiency declines as membrane integrity weakens. None of these effects can be predicted by caloric value because they are not functions of energy quantity. They are functions of material composition. The calorie model also fails to account for time. Structural integration is not immediate. Dietary lipids are absorbed, transported, and gradually incorporated into cellular membranes through continuous remodeling cycles. Each cycle replaces a portion of existing structure with new material. Over weeks and months, this process shifts the physical properties of tissues. A system repeatedly supplied with unstable inputs will accumulate instability. A system supplied with stable inputs will move toward coherence. Energy accounting captures none of this because it treats each intake event as isolated rather than cumulative. Once energy became the dominant framework, interventions followed the same logic. Reducing fat intake was interpreted as reducing energy density, without considering that this also reduces the availability of structural materials required to maintain membrane integrity. Replacing those fats with carbohydrates preserved caloric intake while altering the composition of the system being built. The body continued to assemble itself, but with different inputs, leading to predictable changes in behavior that were then labeled as new conditions rather than recognized as consequences of altered structure. The collapse into calories also created a disconnect between measurement and outcome. Individuals consuming similar caloric amounts can produce entirely different physiological states because their internal structures differ. One system may maintain stable membranes, coherent signaling, and efficient energy production, while another operates with degraded boundaries, erratic signaling, and constant compensatory stress. The calorie model cannot explain this divergence because it does not measure the variables that drive it. Reintroducing structure into the model resolves this gap. Food is not an abstract source of energy. It is a set of materials that enter the body, integrate into its architecture, and alter its behavior over time. Lipids in particular do not pass through the system unchanged. They become the system. They define the properties of membranes, the behavior of receptors, and the stability of energy production. When this is accounted for, outcomes that appeared inconsistent begin to follow predictable patterns. Systems built from stable materials behave in stable ways. Systems built from unstable materials require constant compensation and eventually lose coherence. The mistake was not in measuring energy, but in allowing that measurement to replace the underlying reality it was meant to describe. Calories can quantify potential, but they cannot define function. Function emerges from structure, and once structure is removed from the model, the system becomes impossible to predict with any precision, lipids reclassified as fuel instead of material. Why energy models fail to predict biological behavior. Energy models treat the body as a system defined by input and output, where biological outcomes are expected to follow from the balance between what enters and what is expended. This assumes that the internal machinery behaves consistently regardless of what it is built from. In practice, the machinery itself is variable. Cells do not process energy in a uniform environment. They operate within membranes whose composition determines permeability, electrical resistance, receptor behavior, and transport dynamics. When those properties shift, the same amount of energy entering the system produces different results because it is being processed under different physical conditions. A calorie does not specify how energy is handled once it reaches the cell. It does not determine whether glucose transporters are properly positioned within the membrane, whether insulin receptors can cluster and signal efficiently, or whether mitochondrial membranes can maintain the gradients required for controlled energy production. These are structural variables. When membrane composition becomes unstable, transport becomes inconsistent. Substrates either fail to enter efficiently or enter without proper regulation. This creates oscillation in availability, forcing the system to compensate through hormonal adjustments that appear as dysregulation rather than a predictable response to altered structure. Mitochondrial function exposes the limits of energy-based thinking more clearly. ADP production depends on the ability of the inner mitochondrial membrane to maintain a proton gradient. That gradient is a physical separation held by the integrity of the membrane itself. If the membrane becomes permeable due to lipid instability or oxidative damage, protons leak across it. The system responds by increasing electron flow in an attempt to maintain output, which raises reactive oxygen production and further degrades the structure. Energy is still entering the system, but it is no longer being converted efficiently because the conditions required for controlled conversion are no longer present. A calorie-based model cannot account for this because it does not include gradient stability as a variable. Signal timing is another dimension that energy models do not capture. Hormonal signaling depends on receptors embedded within lipid environments that determine how signals are initiated, amplified, and terminated. When membranes lose organization, receptor behavior becomes inconsistent. Signals may initiate without proper amplification or persist longer than intended due to delayed termination. This produces overlapping signaling states where multiple pathways remain active simultaneously, creating noise within the system. The energy entering the system has not changed, but the interpretation of that energy at the cellular level has become distorted. The model also fails to account for how energy demand is generated. Demand is not fixed. It is influenced by the stability of the system itself. When membranes are compromised, ion gradients leak, requiring constant correction. The cell expends energy not for productive work, but to maintain basic separation. This increases baseline energy consumption without improving output. Two systems receiving identical caloric input can diverge because one is spending energy maintaining unstable structure, while the other is directing energy toward functional processes. The difference is not in the quantity of energy, but in how much of it is lost to maintaining compromised architecture. Time further separates energy models from biological reality. Structural changes accumulate gradually as membranes are remodeled with incoming materials. This alters system behavior long before changes in energy intake occur. A system can move toward instability while caloric intake remains constant because the materials being integrated are shifting the properties of the structure itself. By the time measurable outcomes appear, the underlying architecture has already changed. Energy models interpret these outcomes as imbalances in input or output when they are the result of altered structural conditions that were never measured. Energy accounting provides a narrow view that can describe how much enters and how much leaves, but it does not explain how the system behaves in between. Biological behavior emerges from the interaction between structure and input, not from input alone. Without accounting for membrane composition, signaling organization, and gradient stability, predictions based on energy remain incomplete. The same input can produce different outcomes because the system processing that input is not constant. Once structure is included, variability becomes predictable. Without it, outcomes appear inconsistent and are attributed to factors that do not address the underlying cause. The return to structure as the primary model. Reintroducing structure restores the missing variable that connects input to outcome. The body is not an abstract processor of energy, but a material system assembled from specific components that determine how every process behaves. Once this is recognized, the question shifts from how much energy enters the system to what physical conditions exist inside it. Membranes define those conditions. Their composition sets permeability, electrical resistance, receptor organization, and the stability of gradients that drive transport and energy production. When these properties are consistent, inputs are handled predictably. When they are not, the same inputs produce variable and often unstable outcomes. Structure establishes the rules under which all biochemical reactions occur. Enzymes do not operate in isolation. Their activity depends on orientation, proximity to substrates, and the surrounding environment, all of which are influenced by membrane state. Receptors require organized lipid domains to bind ligands and transmit signals with precision. When those domains lose coherence, signaling becomes fragmented. Inputs still arrive, but they are interpreted differently because the physical context has changed. This is why restoring structure precedes any attempt to correct downstream processes. Without stable architecture, interventions act on shifting ground and produce inconsistent results. The return to structure also redefines how time is understood in physiology. Structural change is cumulative. Lipids introduced through diet are incorporated into membranes through continuous remodeling, gradually altering their properties. This process does not produce immediate shifts in the way energy intake does, but changes the operating characteristics of the system over longer intervals. As membranes are rebuilt with more stable materials, permeability tightens, receptor behavior stabilizes, and gradients become easier to maintain. The system begins to require less compensatory effort to sustain basic function, and energy can be directed toward productive work rather than constant correction. This framework resolves the apparent disconnect between similar inputs and different outcomes. Two individuals consuming comparable amounts of energy can produce different physiological states because their internal structures are not equivalent. One system may be built from materials that support stable membranes and coherent signaling, while another is assembled from components that degrade under stress and require continuous repair. The difference is not explained by energy balance. It is explained by the condition of the structure processing that energy. Once structure is measured and addressed, variability becomes predictable rather than arbitrary. Returning to structure changes how intervention is approached. Instead of attempting to force outcomes through isolated adjustments, the focus shifts to rebuilding the conditions that allow the system to regulate itself. Stable lipid inputs replace unstable ones. Oxidative pressure is reduced so membranes can maintain integrity. Signaling pathways regain clarity because the environments that support them are restored. This does not act as a direct correction of every symptom. It alters the underlying conditions that produce those symptoms, allowing multiple systems to realign simultaneously because they share the same structural foundation. In this model, nutrients are evaluated by what they become within the system rather than by their immediate effects. Lipids are not consumed for energy alone. They are incorporated into membranes where they influence behavior continuously. Some reinforce stability and resist degradation, supporting consistent signaling and energy production. Others introduce fragility, increasing susceptibility to breakdown and amplifying noise within the system. These differences accumulate over time through repeated incorporation, gradually shifting the operating state of the organism. The returned structure does not discard energy. It places it in context. Energy remains necessary, but it is no longer treated as the primary determinant of function. It is a resource that must be processed within a defined architecture. When that architecture is stable, energy is used efficiently and predictably. When it is unstable, energy is diverted toward maintaining basic conditions rather than producing useful work. By restoring structure as the primary model, the body becomes understandable again because its behavior follows from physical constraints that can be observed, influenced, and rebuilt.